Okay.
Today it's my pleasure to talk to someone I met in Fort Collins in the biology building in, I guess, mid-1980s, mid to late 1980s when I was doing a postdoc there. And he studies a neuroendocrine system that controls molting in crustaceans, crabs, lobsters, things that people here in Baltimore, Maryland area, and Maine, and not all over the country because they ship them all over, eat. So there's a lot of interest in control of molting because that's the way these creatures grow. If they don't molt, they don't grow. And so anyway, welcome, Don.
Thank you.
And
it's good to be with you.
So you're professor of biology at Colorado State University, where you're head of the crab lab, which I looked on your website. It's a lot bigger than it was back when my wife, Joanne, was doing a master's degree in your lab back in the 80s. And when did you first get interested in crustaceans?
Oh, ever since I was an undergraduate. So I'm originally from California, grew up in the Central Valley of California, but my family would often go to the coast. I became really fascinated with marine life. It's so bizarre in so many ways. And so that sort of planted the seed in terms of my future direction. So when I decided to go to college and I selected going to UC Santa Barbara and studying marine biology there. They have a marine lab right on campus, and so that was really the main impetus. So it gave me access to experiencing and observing all the sorts of marine life that occur off the coast of Santa Barbara. And while I was taking an invertebrate physiology class, back in the day, it wasn't really common for an undergraduate to get involved in research. But what was really innovative about Santa Barbara was professors created these courses that actually introduced students to research. And so we had to settle on a particular project. I got interested in a crab species that's found in the waters off Santa Barbara, and I started doing some physiological studies, and I really became fascinated with these animals and knew that I wanted to continue whatever postgraduate training to engage that kind of research on crustaceans. I was originally interested in kind of osmoregulation and ion transport, and so I applied to UC Berkeley in the zoology department there, was accepted, and became interested in pursuing this in crustaceans and working with one of the zoology faculty there, UC Berkeley, Ralph Smith. And Ralph was really kind of old school. He said, you don't have to do anything really associated with what I do. I want you to pick a project that you think would interest you. And so for a couple of years, I sort of explored different aspects of crustacean physiology and became interested in molting physiology, particularly the process in which animals molt, and particularly how they can drink huge amounts of seawater to increase the pressure that inflate the new exoskeleton. So they, as you say, crustaceans have to molt in order to grow, and basically because of the hard exoskeleton they have. It's with arthropods, it's extremely successful body plan, right? You've got now an outer shell that can serve for mechanical support, but also protection. And but one of the problems with having an exoskeleton is that it restricts growth, and you have to practically shed that exoskeleton, make a new exoskeleton underneath that's larger. And so you have to extricate yourself out of the old shell. And then once you do that, you can drink copious amounts of water to increase the internal pressure, stretching out the new exoskeleton to create more space. And so it was, and so I became interested in sort of this process. How does an animal do that? How can they drink half or more of their body weight in water and transport that water from the gut, from the stomach area or the gut into the blood space, right? And that's what you have to do to make more space inside. And it just so happened that at the time, at Bodega Marine Lab, which was operated by UC Berkeley at the time, there was a major aquaculture project at Bodega Marine Lab, where I'm currently working right now, studying or developing the technology for raising East Coast lobsters in a completely artificial environment, right? A closed system. And that gave me access to huge numbers of juvenile lobsters where I can actually study the very short time, maybe 3 or 4 hours, in which these animals imbibe water, shed their exoskeleton, and continue to imbibe and move that water across the hemolymph. And so that became sort of my really entry into molting physiology. And that was back in the 70s, right? When I was a graduate student, I spent a great deal of my time working on that in the mid-1970s. And that pretty much determined the trajectory of my career since that time. So I graduated with my PhD in 1979. And so that's the short story.
And you've studied a number of aspects of what happens during molting. When Joanne and I were there in Colorado, you were doing a lot of work on the muscles, because you want to explain why the muscles are. You explained how they.
Oh, sure.
That's also a dramatic process where the muscle breaks down rapidly.
That's very true. So after I received my PhD, I was fortunate to do a postdoc position at Oak Ridge National Laboratory with, again, a major influencer of crustacean physiology, Dorothy Skinner. She was a scientist there. And so when I approached her, she's in my interest in molting physiology. She says, one thing we've discovered many years ago, but really haven't pursued, is this process in which the claw muscle undergoes a dramatic atrophy. Now, as most people know in this program, when you have a crab, they usually have very large claws. And a problem is a mechanical one. When they actually have to pull the claw out at the time they molt, they have to squeeze this large distal claw through a very narrow joint that connects the claw to the body. And the only way they can do this is actually to reduce the muscle size. And she found that they would atrophy at least 40% of their muscle mass in a matter of weeks. And then they would grow this muscle back after they successfully molted. And she said, this is a really fascinating problem, but we, as I said, we don't know exactly how this works. What are the mechanisms involved? What are the enzymes that are important for this process? And so I came to Oak Ridge to study that. And that's really determined my research for the next decade or two. And that was our focus when I came to Colorado State in 1985, established my lab, and that was the primary focus of our research at that time. And Joanne was a master's student at that time and contributed to that.
And these dramatic changes you described in the accumulation of water so that the crab can get bigger right after it molts, so that when it lays down a new skeleton, it's bigger, and then this dramatic process of breaking down muscle, and those are all controlled by a hormone, right? Those processes. You want to talk about the molting hormone?
Correct. So, sure. So in terms of the water uptake, and this is not really work that I've done, Simon Webster at Bangor, Wales, and others identified neuropeptides that are produced, interestingly enough, in the gut itself that actually promote this large water flow. And there's a spike of these peptides that trigger this event and facilitate the movement of this large amount of fluid from the intestine into the hemolymph. What we focused on primarily the role of steroid hormones that coordinate all these physiological processes, and these are steroid hormones, as I said, based upon the molecular structure of cholesterol. So the same kind of hormones that are found in mammals that are based on cholesterol, like testosterone and estrogen. Crustaceans also have these steroid hormones that are unique to arthropods, but essentially coordinate all these physiological events. So claw muscle atrophy, synthesis of the exoskeleton, and so forth. These are called ecdysteroids, and they come from the word ecdysis, to shed. That's the Greek word. And so that's where these are molting hormones, and distinguish these hormones from other ones, such as in mammals. But these ecdysteroids really are important, produced by a pair of endocrine glands in the body called the Y organ. And so these are in the body of the animal, and these Y organs synthesize and produce the ecdysteroids, and those Y organs in turn are controlled by neuropeptides that are produced in the brain and the ganglia in the eye stalks. And one of these hormones, peptide hormones, is called molt inhibiting hormone. As you know, Mark, you did your PhD work with Gene Spaziani in Iowa, and you studied using pharmacological approaches primarily the signaling pathways that are controlling the activity of the molting gland or Y organs. So you and I have a lot in common, even though we've kind of moved paths. But it's kind of fun to be able to relate our work with what you did as a PhD student back in Iowa.
This, one way to look at this from an evolutionary perspective, and the way that's kind of doing back in my PhD work, was kind of say, what, is this kind of, first of all, that molt inhibiting hormone, MIH, is produced by neurosecretory cells, and they're kind of similar in a sense to the neurons in the posterior pituitary gland of mammals, that
exactly
they secrete into the blood neuropeptides like vasopressin or oxytocin, and but then really the intriguing thing, you described how these events of molting itself occur relatively rapidly in relation to, like, the normal life of a crab. They only occur intermittently during short time periods. Then there's long period where they're not molting. And the intriguing thing to me, I think to a lot of us, is that this molt inhibiting hormone, during all this time that their animal is not molting, it's being released into the blood of the crab and preventing the production of the molting hormone.
Yeah, it's a really wacky system. When I'm training students and I tell them this, they look at me really puzzled. And they first of all say, "Why does that happen this way?" And I'm sorry, I can't. Those why questions are often our guesses. We have no idea why this is regulated in this way. An interesting bit of trivia, Mark, is that the neurosecretory system in the eye stalk was actually the first description of that kind of process in any organism. And this was back in the 1950s. So before anyone knew about the neurosecretory mechanisms in the pituitary gland or other types of systems, the the description of this system, it's called the X organ Y sinus ganglion complex, but these are neurons that are neurosecretory. They are controlled by the ganglia and the eye stalk, and ultimately control the release of MIH from the sinus gland into the blood space of the crab. As I said, this was a novel discovery back in the 1950s, and now we find it quite common in all sorts of different types of animals.
And back then I was thinking that maybe what's happening is, well, it's very important that when a crab or lobster molts, it's like in a safe place, because during that time it doesn't have a shell to protect it. And so I was thinking, well, maybe like normally the crab's life is fairly stressful, and then like when it finds a place it can molt, then its stress levels go down. So I was kind of thinking this like the stress responsive hypothalamic pituitary adrenal gland system. You know, so I did these experiments. We had a marine room like you do, right? And so the crabs I studied, Cancer antenarius, they're marine crabs, and so we had a marine aquaria, and so I went up there every day, and for several hours every day, I stressed the crabs by irritating them with these long. I just poke them and make them mad for a long time period, and then using our bioassay back then, and this is something I want to know about, because about what's the amino acid sequence of this MIH. But anyway, that wasn't known back then. But we had this bioassay for MIH where we take extracts of the eye stalk and then expose cultured Y organs to the extract, and then all I did was radioimmunoassays. I measured the disteroid production level released into the medium, but I found that when I took the eye stalks from the stress crabs, there was more ability of that extract to inhibit the disteroid production. So, you have any comments on that?
Well, so now in hindsight, given 4 decades of research by myself and others, is that we now appreciate that the control of molting is extremely complex. It's clear that the animal is integrating both external and internal cues to determine when it is the best time to molt. Okay, and so things like, we know that stress, we know that crowding inhibit molting. So clearly that supports maybe the continued secretion of MIH to prevent those animals to molt. But we also know that there's internal cues. So there's constant communication between the eye stalk ganglia, the Y organ, and other organs to determine when internally the animals have the necessary energy stores, that the tissue growth has reached a point where it's filled the new space. So now the tissues can't grow anymore because they've filled the internal space. So those kinds of things. But ultimately, the trump card is MIH. There are other signals that are coming in, but the master hormone that controls this is MIH. So even if the animal is receiving signals from the outside that or internally that the tissues have grown to sufficient strength, but if the environmental conditions are not, it could be photoperiod, it could be temperature, it could be a shelter that they can have access to, all that is weighing into that decision, because obviously, as you know, when an animal is molting, they're extremely vulnerable. And that's probably the most critical part of any crustacean's life history, is the ability to molt successfully. And And so there's this essentially this drive to grow, but at the same time, they're not going to stimulate that growth if they know that the conditions are not optimal for them to do that. Does that make sense?
Yeah. And
But
and
it is, but it is
and so now my understanding is the amino acid sequence of MIH has been established. Does it have any similarity to any mammalian peptides, neuropeptides?
no,
It
no.
does.
Then this neuropeptide family that includes not all, and these are a number of peptides that are produced in the eye stalk ganglia that control a variety of different functions. You mentioned the similarity with the pituitary gland. When I explain how molting is regulated in a crustacean, I say, well, you can think of the X-organ sinus gland complex as the crustacean's equivalent to the pituitary gland.
Okay.
It seems to be one of the master endocrine glands that control a variety of different physiological functions. So MIH, of course, controls molting, but also it controls reproduction. It controls the growth of hormones of gonads, like the ovary, in terms of oogenesis, creating eggs that can be fertilized. It controls metabolism. So there is another neuropeptide that controls the utilization of carbohydrates and glucose in tissues. So it's obviously not as complicated, at least we don't know. I don't think it's as complicated as the pituitary gland, but it's analogous to that. So organisms need some sort of endocrine system that controls really important physiological processes.
Yeah.
And it seems to be neurosecretory mechanisms and these neuropeptides that control those various processes. But yes, the number of MIH genes have been cloned and sequenced in a wide variety of decapods. Same with these other neuropeptides. Genomes have also helped with that. Transcriptomics has also helped with that. So we know, and structural studies indicate that these are very interesting polypeptides. They're called neuropeptides, but they're really big peptides. They're on the order of like 78 to amino acids in length. So it's not like many peptides are small, usually less than a dozen amino acids. So, but they have a very distinct structure, compact structure that's controlled, that's stabilized by disulfide bridges. And that really moves us into kind of our current area of research. But we can get to that in a moment.
Yeah.
But I think getting back to sort of this curiosity thing about why does the animal, a major group of arthropods, produce a neuropeptide that inhibits molting, which means, as you point out, the intermol period in which the animal is actively engaged in foraging and mating, blah, blah, blah. They're not in the process of either preparing for molt or molting or recovering from molt. This can be months or even years, depending upon the species and the environment and everything else. And so this seems kind of stupid, because that means it has to continue producing this neuropeptide constantly for months or maybe even years, right? Yeah. So that's one of those enigmas that we were working on, and led us to a hypothesis that explains that you can keep the Y organ inhibited even if there's a pulsatile release of MIH from the Neurosecretory center. So but we have a model in which we can explain how MIH can inhibit the Y organ from making a disteroid for long periods of time and not using a whole lot of neuropeptide to do it. Does that make sense?
Yeah.
So, but what it's interesting is that when you look at insects, okay, here's another major group of arthropods, and they have a stimulatory system. So they produce a neuropeptide. It's not related to MIH, but it actually is produced when the animal wants to molt, which makes more sense to me, right? And so now we have this really interesting situation where we have two major groups of arthropods, both have to molt, but the regulatory mechanism is completely opposite. It's weird.
So can you talk about?
I can't explain it. It's just the way it is.
Yeah. Can you talk now about what's happening in the Y organ? How is MIH inhibiting the disteroid production by the Y organ?
Mm-hmm. Well, so the neuropeptides are large, polar, and so they cannot cross the membrane to transmit a signal to the inside of the Y organ cells, right? So this is called signal transduction, where a neuropeptide binds to a receptor on the surface of the membrane. That induces a change on the opposite side of the membrane, leading to a chain reaction of proteins that are transmitting the signal to the actual synthetic machinery of the Y organ. And as you know, you've done a lot of this work back when you were a PhD student, is that this is mediated by cyclic nucleotides, right? Cyclic AMP and cyclic GMP. And there's certainly some variation between species, but basically we all agree that that signal is transmitted by cyclic nucleotides that are actively changing metabolic activity of enzymes that alter the secretion, the synthesis and secretion of molting hormones, right?
Yeah, and I think Don, I think it's a good point to interject here. People may be asking, and eventually maybe at the end of this we can talk a little bit about what. So these things you're studying are clearly important to crab populations. There's, climate change. There's a lot of factors affecting these organisms, and we'd like to have them around a long time. But studying these simple animals are evolutionarily more ancient, is of great value just to basic science. And there have been so many discoveries made in lower organisms, whether it's insects like Drosophila, worms like C. elegans, or you mentioned cyclic AMP. Eric Kandel got Nobel Prize for his work in a sea slug, which is essentially a marine snail that doesn't have a shell. And cyclic AMP playing a critical role in learning and memory. So this system that of cyclic AMP, calcium cyclic AMP, plays so many important roles in so many processes. But anyway, maybe 20, 30 years from now, your work there'll be something related to these mechanisms of molting that has significance for, well, I think it already does, but for humans beyond just eating the crabs or lobsters.
Thank you. That's very kind.
Okay. So
no, but these, you're right. These signaling pathways are highly conserved. So just because you have a crab doesn't mean you've got an entirely different system than applies to mammals. So you're right. What we learn from Drosophila and other models, which are easier to work with and more ethically, available to work with, can inform more complex, perhaps, or particularly maybe even some human disease aspects, neuropathologies and so forth, understanding those processes, because biology is terribly conserved. And I mean, there's nothing really majorly innovative in biology. Evolution works on existing systems, and so often various processes are co-opted for completely different purposes, but the basic mechanisms are the same. Nature has to work with what's there. It can't just sort of de novo make something new. That's very unusual.
And you've been able to you mentioned the word complexity. Well, it turns out that the the genome of crustaceans, the ones you've looked at, are quite complex, right? Even in comparison to human, they're at least as complex, I think, in terms of
Yes.
number of genes and so on.
Yeah. No, it's we in the Y organ, with our transcriptomics, we've identified. So with RNA sequencing, this high throughput sequencing, the technology that's now available, we've been able to take advantage of that. And basically, we wanted to catalog every gene that's expressed in the Y organ over the molt cycle. And we have around 38,000 unique transcripts that are found in the Y organ. So that's on the order of what you'd find in any human tissue.
Yeah. and you've spent, so you sent me a few articles before this, one where you kind of reviewed this, gene expression analysis, where essentially you're looking at the messenger RNA encoded by genes.
Correct.
And all of them in the Y organ cells. And you've discovered some very interesting, we talk about conservation of mechanisms and pathways, and so you've discovered a number of interesting pathways that are known about in mammals, and we can talk about this mTOR pathway in particular, but why don't you kind of describe some of the key pathways? You mentioned cyclic AMP, maybe calcium, and then there's transcription factors that induce genes, but you find, these are very extensive studies. You've looked at this at different stages. You mentioned these different stages of the molt cycle, intermolt, early in molting, when you get this big change in water accumulation, et cetera. So can you, it would take many hours for you to describe this in detail, but could you kind of highlight some of the most interesting discoveries you made in that work?
Happy to. So let me just give you a little bit of perspective. So throughout my career, what I've tried to do is exploit new technologies in terms of advancing our research, right? So so a lot of my work early on was protein biochemistry, characterizing enzymes that break down muscle proteins, blah, blah, blah. Joanne had a big part of that in terms of her master's thesis. Then we got into cloning back in the early 90s because I wanted to get a better, more fundamental idea in terms of at the transcriptional level, what genes are involved in characterizing those. And then PCR came along, so we were able to advance that further with terms of identifying and pulling out genes of interest. But this was largely a gene by gene approach, and we had to work with existing knowledge. So we'd say, we think, protein kinase A is involved. So let's look at the database in GenBank and pull out sequences from other organisms. We'll do alignments and identify highly conserved regions, and then we'll make primers that will amplify that particular region, blah, blah, blah. It was a very painstaking and tedious process, as I'm sure you appreciate in terms of your own research. And
you have to you make educated guesses and hope that, you get lucky and find something interesting.
Exactly. So you're investing months really doing this, and hopefully it pans out, right? But the real drawback of that is that you only can base this on existing knowledge and existing data resources, right? And so what always bugged me, or at least not bugged me, but just made me really uncomfortable, was that we were unaware of anything that that might be going on because we just didn't know it existed, right?
Yeah.
And so when high throughput sequencing came along back, in the early 2010s, we saw an opportunity because we didn't have a genome of the animal we studied, which is mostly a terrestrial crab that we study in Colorado. More about that later, but this has proven to be an excellent model for studying various processes of molting, and this same species is what I was introduced to when I worked with Dorothy Skinner at Oak Ridge National Lab. That was her primary experimental organism. But so here we had an opportunity to be able to not, wait on a genome, because that was going to be years, from then, be able to kind of skip that and actually be able to sequence every gene transcript that was expressed in the tissue at a particular time. I tell my students, be careful what you wish for, because it revealed an enormous complexity of the genes that are expressed in the Y organ and how they're changing over the molt cycle. As I said, we cataloged about 38,000 unique genes, right? Okay, that's a lot. What that revealed is that certainly it showed that there's the signaling pathways that we knew about in terms of MIH, right? With cyclic nucleotides, we found all those genes, transcripts, and everything else, and they were changing over the molt cycle. That wasn't a surprise. But we identified, I think it was about maybe a couple of dozen different signaling pathways that were expressed in the Y organ. And then characterizing the receptors that are, so we want to know what the MIH receptor is. We still haven't really identified it yet, but given the fact that it entails these cyclic nucleotide messengers, that generally is in pathways that have G protein coupled receptors in the membrane, and so these are important for binding a signal like a neuropeptide, transmitting that to the cell. So, but and these receptors have a pretty unique structure. They're single polypeptides, but they have these 7 transmembrane domains that go in and out of the membrane. So that makes them pretty unique, pretty easy to identify. So when we looked for transcripts that encoded these 7 transmembrane regions, we identified 99 different G protein coupled receptors. And about 2/3 we could, with homologies to other systems, we could identify what the potential ligands were, but another third, we had no idea what their ligands are. So what that means, Mark, is that Y organ can detect up to 99 different ligands. And transmit those signals to the cell. So that means the Y organ is at least potentially capable of responding to dozens of different signals that are coming from maybe different tissues or from the environment, whatever. So it's so obviously it's revealed a wealth of information. So now we have at least we now have a pretty good idea of the complexity of the system. Now it's a matter of figuring out what each is involved in that.
Now, I guess we didn't mention, Don, this is kind of important from a practical standpoint. Your students and postdocs in your lab, they don't just wait around until the crab starts to molt. They can cause it reliably to molt.
Correct. Yes. When you study molting, you've got to have a good model organism in which you can maintain them easily in the laboratory, and we can also induce molting. We can do various treatments to control these animals in molt. Because you're right. NSF doesn't understand why you haven't published anything, because the animals haven't molted in 2 months. So you can't just sort of wait around and wait for them to spontaneously molt. So as I said, we work with a terrestrial crab. It's called the blackback land crab. We get these animals from the Dominican Republic. They're shipped to us from there to Colorado. We typically get about 800 animals. In fact, we're going to get another shipment of animals next week. And then by air cargo, and then we'll take them back to our lab and get them into our environmental chamber. So these animals are easy to maintain in the lab. As you mentioned, you worked with a marine species in Iowa. Maintaining marine aquatic systems is really time consuming, right? And you have fairly limited space to maintain marine animals in Iowa or any place away from that. So these animals, because they're terrestrial, so this species lives throughout the Caribbean and the islands of the Caribbean near the beach levels, usually just above high tide level in the coral dunes with vegetation and so forth. They dig burrows in the substrate.
I might have got pinched by one of them. Joanna and I went to St. John's Island, and
okay.
Yeah, and there's these land crabs, in little holes in the sand. They weren't really close to a body of water, though, or at least that we saw.
Well, there's this particular species is found close to the beach, but there's actually a related species that's more inside in the more forested areas of that. And there's also a large land crab that's often popular with people there to eat. They get large enough, so but this particular species is easy to collect because you go out in the early morning, they're out there feeding on the substrate, especially after a rain. And so you can just run with a bucket and grab them and throw them in the bucket to sorting it and ship them out. But so terrestrial means that we don't have to have a seawater system, right? They don't need standing water at all. They get their water primarily from their diet. So they breathe air. They just have to be maintained at a constant temperature of around 82 degrees Fahrenheit. That seems to be optimum at high humidity. And then we feed them, lettuce, carrots, and raisins. They love raisins, by the way. But this provides a very good diet for them. The lettuce primarily, it doesn't have much nutritional value, but it's high water content. And so that's where their primary source of water is from their diet. So the land crabs are not really picky eaters. They're omnivores. So there's some crabs that are strict carnivores, and that is more of a problem in terms of providing that kind of diet. But as you mentioned, so they're easy to maintain in the lab. Just have to keep them under optimum conditions that mimic the tropics. They use their burrows primarily for protection, and they use that as a shelter for when they want to molt. They go down in their burrows and molt. The mating occurs in their burrows, or when the surface conditions either is too dry or too hot during the day, they'll go down into their burrows and wait it out until usually dusk to go out and feed again. And that's, of course, that's a refuge for predation.
So in their natural environment, if it can be harsh, or they can be fighting between crabs, or maybe some other species grabs a crab and breaks off or amputates a couple limbs, and that's a trigger for molting. Is that what you're using?
Yeah, that's one of the main ways we induce molting. So crustaceans, if they lose an appendage like a walking leg or claw, they can actually regenerate that appendage, which is really pretty clever. The problem is that these animals have to molt to regain the function of that regenerated leg. So, for example, they'll grow a limb bud, which is a fully regenerated limb, but it's all coiled up into a cuticular sac, and so that leg has to be extended when the animal molts. So when they, as I said, when they increase the internal pressure inside, stretching out that new exoskeleton, that also extends these regenerates. So this forces the animal that if they want to get that functional leg back, they have to molt to do that. What Dorothy Skinner and her students discovered back in the 60s was that if these animals lose 5 or more walking legs, that can actually stimulate them to molt. Your viewers might not know this, but these are decapods. That means they have 10 feet. The first pair of appendages is the claws, and then it has 4 pairs of walking legs. And so, they've got a lot of legs to work with, right, to maintain mobility, and they're quite good at adapting to loss of a leg or two. But when you start losing 5, 6, or maybe all 8 walking legs, the mobility is severely constrained, right?
Yeah.
And particularly with terrestrial species, because they don't have the neutral buoyancy of water to maintain the body, it's even more imperative for them to regain the functionality of those legs. So what we typically do is we'll induce these animals to autotomize these legs. So this is really an interesting process. So autotomy is actually triggered by the nervous system, by the animal. It's a conscious act that the animals do to escape predation. So for example, a bird grabs a walking leg of a crab, and so what the animal can do is autotomize that leg, drop that leg, leaving that leg in the beak of that bird, and they can then scurry down into their burrows. And then it's from that base of the leg that they'll form a new limb bud and grow it back. So as I say, we don't remove legs, we actually induce that autotomy reflex. And we do that by just essentially doing a little crimp on their knee of the leg. Oh. And what's interesting too, we know that it's a conscious act by the crab, because if the animals have to think that they can escape if they're going to drop that leg. Oh. So they have to be in contact with the substrate. And if you crimp the leg just slightly, they'll drop that leg immediately. Huh. If you hold the animals so they can't escape, and you do the same kind of treatment, they won't drop their leg. So this is actually intentional and inducing a reflex that the animal is making this decision that is it better to lose a leg and live, yes, or keep the leg and die. Yeah. Okay. Yeah. But as I said, if they lose a leg or two, it's not a big deal. They still are quite mobile. That won't stimulate molting. But if they lose at least 5 walking legs, obviously their mobility is impaired, and that is sufficient to induce molting. At that point, we remove them from the group cages and put these animals in their own individual cages. Yeah. But that's really our main way of inducing molting. And then we can just monitor the size of the limb buds of the animals, and because when they enter pre-molt and respond to these rising levels of ecdysteroids in their blood, those limb buds will start growing. And so we can actually follow the progress of the pre-molt period when they're advancing toward ecdysis. And so we can, within fairly good accuracy, we can actually monitor and expect when these animals are going to molt. So we can sample tissues from pre-molt animals. We can let them molt and sample the wire-ins from post-molt animals to understand these changes in gene expression. So yes, so for us, it turns out to be a really good model system for us to study molting processes.
Can you talk about that? One of the pathways that interests me, and well, all these do, but I'm interested in everything, but is a mTOR pathway. We've done a lot of work on effects of fasting and caloric restriction, mainly on the brain in terms of neuroprotection. And then, but so I think the intermittent fasting is a good kind of illustration. Maybe we can kind of relate that to MIH acting on the Y organ and MIH, there not being any MIH to act on the Y organ. So with fasting, essentially the cells go into a kind of conserve resources, you don't grow. And so this mTOR pathway is activated by nutrients outside the cell from the blood. So if you eat a high protein meal that's broken down to amino acids, then your blood amino acid levels go up. And then that will activate the mTOR pathway. And it kind of makes sense because you just eat something, you want to get amino acids into the cells so that they can grow. But during fasting, mTOR is turned down, and another process called autophagy, which is a cellular garbage disposal and kind of recycling as well, system goes up. So that has to do with the conserved resources. Again, you can kind of recycle the amino acids from proteins you already have or whatever. But you discovered one of the main pathways in your gene analyses pathway when you look at different stages of the molt cycle was mTOR.
Yep. Yep. No. Yeah. So, as you know, Mark, mTOR is a very important enzyme that controls energy utilization in cells, and mTOR is ubiquitous. It's found in every eukaryotic cell, animal, plant, fungus, everything, and it's highly conserved. It's a huge protein. It's like 2,500 amino acids. And it's got all these multiple domains and functions, including protein phosphorylation, and that's how it transmits a signal to control protein synthesis. But yeah, it's remarkably conserved. It's probably the most important protein that no one knows about. And I don't really understand this because, as you know, this is so important in terms of how the cell responds to a variety of different signals that affect synthesis of proteins and other molecules, right? Having nutrients like amino acids, you've got to have the building blocks of proteins to actually synthesize proteins, right? So that's so mTOR is stimulated by nutrients, particularly amino acids, that say, okay, I've got enough building blocks to synthesize protein. So I'm going to stimulate protein synthesis, right? But you also have to have nutrients to provide the energy necessary for doing that, right? So, and stress, we know, inhibits mTOR activity because, again, if cells are under stress, they have to conserve energy and maintain cellular functions elsewhere, use whatever available energy is there to do that. It's hypoxia, low oxygen levels will inhibit mTOR. Growth factors stimulate mTOR. So, in fact, one thing that's fascinating about mTOR is that it's often associated, elevated levels of mTOR are associated with a whole variety of different cancers. Yeah. It's also been implicated in a number of neuropathologies like Parkinson's, epilepsy, even autism. So it's like, okay. And so it's really baffles me why people don't really know about this. It's like I tell my students that to try to emphasize the importance of mTOR is I tell them, whatever the question, the answer is mTOR.
That's Don. I think people are getting more interested, not because they're necessarily interested in the science, but because they can buy a chemical that inhibits mTOR, and there's this evidence. So in rats and mice, daily calorie restriction, every other day fasting can extend lifespan a lot if you start it when they're young. The NIH,
well, even in Drosophila,
and Drosophila,
even in Drosophila,
NIH funded, this is a long time ago, they started this where they gave a lot of money to 3 different centers. Yeah, I see we've been on an hour. Do you have to leave at a particular time, or?
no, I'm
Okay,
fine.
okay.
You want to continue this?
And but anyway, and there's kind of these random studies coming out, say if they give chemical X to mice during their lifespan, they live longer. But a lot of those couldn't be reproduced. And so they started this 3-center study where all 3 centers use the exact same chemical, whatever, resveratrol, curcumin, all these things that.
Oh,
and the only ones,
right.
the only,
The
you know what the only one was that extends lifespan?
no, what was it?
mTOR, a drug that inhibits mTOR, rapamycin. Right?
Rapamycin.
Yeah.
Right. Yeah. So in fact, maybe we should tell your viewers what mTOR actually stands for, and that's it stands for mechanistic target of rapamycin.
Yeah.
So the story of rapamycin is really quite fascinating. So back in the 70s, there was an expedition that went to Easter Island to take samples, soil samples, with the idea of kind of mining or possible microbes that might be producing antibiotics or other substances that could be of biomedical importance. And so they took a sample from Easter Island, and they grew up these different microbes, and they discovered that one of these strains of soil microbes produced a compound that inhibited growth of yeast cells. And so this drug was called rapamycin. So it's an antibiotic, right? But it's derived from the native language, native name for Easter Island, which is Rapa Nui. And so mycin means antibiotic, and so they named this compound rapamycin. And later discovered through mutation analysis that there was a gene that if you mutated it was resistant to rapamycin, and they ultimately, and they called this gene the target of rapamycin, or TOR. And originally, I think it was so that's the origin of the name. mTOR, the M has stood for a variety of different compounds as it was discovered that it's found in more and more organisms, right? So now I think everyone's settled on mechanistic target of rapamycin, and so mTOR is in Drosophila, it's in mushrooms, it's everywhere. So that's and it's also kind of a commentary, sort of how arrogant science was back in the 1970s. The idea that they could go to Easter Island, take a bunch of samples, and take them away, and without any benefit to the native population in terms of genetic aspects of it, right? We're operating much more ethically these days, but this was back in the 1970s. So the idea of being able to just have a group of scientists go out and just take a bunch of samples and take them back to their labs was sort of the operational norm. There was no thought that maybe they should have share this with the native population.
Yeah.
So and interestingly enough, we have an agreement with the Dominican Republic government that any benefits or applications from our studies using the blackback land crab, they would have the first rights to that. I don't think we'll find anything that's going to be any great of economic importance, but they and other countries have realize that they need to protect their genetic resources, and any applications they should get the benefit from, right? So I think this is a good thing, but it took a long time in coming.
So Don, do you want to describe how mTOR and Y organs changes during the molt cycle, and then what you found when you actually treat these crabs with rapamycin?
Right. So rapamycin is, as you say, readily available. It's a natural compound that's commercially available. And so we use that as a tool to study the effects of rapamycin on the production of the diastроids in the Y organs. And we've done in vitro, that means we've isolated Y organs, cultured them in the presence of rapamycin. And when we do that, we can inhibit the secretion of ecdystroids into the media. We also can inject the compound into animals that are going to molt, and we can inhibit that by this compound. So what's very important is in terms of the control of molting is that when MIH is stopped being released, the Y organs now become de-repressed. They become activated, and they start synthesizing and cranking out ecdystroids, right? And we found that mTOR is critical for that process. So mTOR is controlling protein synthesis, right? And that protein synthesis is necessary for secretion of ecdystroids. And what's kind of fun that we found was this, of course, this goes back to some of your work as a PhD student, where you studied different types of compounds that inhibited protein synthesis, right?
Yeah.
Remember those back then?
Very crude experiments.
You found that, well, that's what we had, right?
Yeah.
But it's nice to be at the point where you can go back and look at those earlier studies, right? And actually make some sense out of it, right? Because admittedly, the tools were available, were limited, maybe rather crude, but you found that that inhibitor of proteins translation was effective at inhibiting ecdystroid secretion in the cancer antennaris Y organ, right?
Yeah, that's right.
And so we know that mTOR controls translation of messenger RNA into protein. So looking back at that, it makes perfect sense that mTOR is one of those primary regulators of protein translation, and complements what you and others have done in terms of looking at the effects of reagents that inhibit protein synthesis. So like cyclohexamide is one of those compounds, right? And so we find that one way to inhibit molting basically is to inject animals with rapamycin, because the Y organs don't become activated, and of course that keeps them from transitioning into other phases as they go through the pre-molt period. So, but I think what's fascinating is that you have a highly conserved signaling pathway that is being co-opted for a particular purpose in a particular organ, like the Y organ, right? Yeah. mTOR has other functions in the claw muscle, and so mTOR is also involved there as well. So it's but different tissues have used this pathway, the same pathway, in different ways for their own purposes, right? For particular process.
Now, you mentioned, you like to apply new technologies to your work, which you've done. What about, can you knock out individual genes or overexpress individual gene in crustaceans in general? Does CRISPR, it should, right? Does CRISPR work or not?
It does work. Limited. The technology is still being developed and applied to crustaceans. There's been some published work showing some degrees of success in terms of knocking out particular genes with using CRISPR-Cas9 technology. But most decapod species are really not conducive to that because they have such a longer life cycle. And this is something that I regret that probably won't be able to take advantage in terms of my work because I'm going to be retiring in a couple of years. But I think what I want to do is provide the resources necessary for taking that to the next level by other investigators that can apply this. But you have to pick the right organism, and I published a paper with Jerome Wee about proposing that a particular freshwater shrimp species might be conducive to using gene function technology. So this is actually a popular aquarium species. It's called the cherry shrimp, Neoparidina, and it has a very, relatively speaking, a very short life cycle. So within about 3 months, they go from a fertilized egg to a reproductive adult, which is really pretty remarkable considering most decapods will maybe 2-3 years to reach that, right? And of course, and it's fresh water, so it's easy to maintain in the lab. As I said, they're very popular with people who have freshwater aquarium systems. They're fun to watch. They swim around, but they're very small. They're only maybe maximum of maybe half an inch in length, overall body length. But the advantage of this is that the species has a direct life history. That means like most freshwater crustaceans, they don't have a larval stage that's free swimming. Those stages occur within the egg, so they have large yolk-filled eggs that maintain embryonic and larval development, and they hatch out as little shrimp, and they'll continue, and they molt rapidly, so they grow very fast. So the advantage of that is that you've got to have a species where it's going to be, if you have big eggs, they're easy to inject with constructs that will target genes with CRISPR-Cas9. Right now, we just haven't had the time or the resources to actually develop this model and show that. But I think that might, right now, we don't have any. The only tool we have for looking at establishing gene function is using double-stranded RNA, where you knock down the messenger RNA of a particular gene, but we don't have anything that will actually do the opposite, stimulate the expression of a gene to overexpress that and see what effect it has. Also, double-stranded RNA is not targeted to a particular tissue. It's part of, it takes advantage of sort of the crab's ability to recognize viral infections and neutralize viruses. So it has a very robust innate immunity system that is mainly functions to control viral infections. But that's about the only technology we have. It's not really, it's not very sophisticated. Let's say that. CRISPR-Cas9 is the tool to use.
Now, do crustaceans, now I'm not aware that they do they ever develop cancers?
Not that I'm aware of. I don't think they live long enough.
Yeah, but
if it's going to,
I don't know.
Out through accumulation mutations, but no, there's no really documented evidence that they have cancer.
But
they don't. Most crabs maybe live a few years. They die of disease or predation or something like that. So
do you know the naked?
So you can't.
Do you know the naked mole rat story? With they apparently don't get spontaneous cancers, and the last I heard, it had something to do with they have really high levels of hyaluronic acid outside the cells. But so I guess my point is, if they don't develop cancers, that might be an interesting species to figure out why, you know. Another thing I was thinking about,
Yeah.
so I did a podcast with Glenn Telling. I don't know if you've come across him at CSU, but he works on prions and wasting
Yeah,
disease.
I know of him. I do.
Yeah.
Mm-hmm.
Do you know?
Yep.
I assume, I don't, I guess I don't know. Do you know if the crustaceans in their genome, do they have a cellular prion protein that's, you know, homologous to the mammalian?
I'm not aware of any prion
Okay.
genes or in our
Maybe in
arthropods.
your remaining 2 years before you retire, you and Glenn can see if the crustaceans are resistant to prion disorder or something like that.
Might be let's see, Glenn might be a hard sell.
Yeah, no, I know. Okay, last couple of things before I let you go. So
Okay.
soft shell crabs are a delicacy, right? Are there like,
Yep.
is there an industries that induce molting by either limitotomy or removing eye stalks, or do they, you know?
Wow. Yes, not in blue crab, which is on the East Coast.
Yeah.
They were, but the crab fishermen there are very good about looking at signs, changes in the exoskeleton to predict when the animals molt. You can pull them out and put them into bins, and just basically wait for them to molt and immediately harvest them and ship them off to restaurants. Right.
Yeah.
I am aware, particularly, so Asia is a big market for crustaceans, and particularly aquaculture. So the white-legged shrimp, for example, the Chinese mitten crab, there's a swimming crab. All those are being actively raised in farms in China and other Southeast Asian countries. So there's enormous demand for crustaceans. These countries, particularly the People's Republic of China, recognize that crustaceans are really a high value source of protein, right? And they're also very popular in terms of those populations and their cuisine. So there's certainly some efforts to, I know, for example, that some shrimp, if you remove one of the eye stalks, that can actually stimulate reproduction. Because again, one of the peptide hormones that are produced in the eye stalk is a gonad inhibiting hormone. So again, reducing the production of that can actually stimulate reproduction. Of course, you want them to produce more of the next generation. They've toyed with multiple leg autotomy, but that's very all these operations are highly labor intensive. I mean, you got a pond, thousands and thousands of white-legged shrimp. You can't possibly do that. So where I think what we see is that particularly China is really investing heavily in technologies manipulations that can use genetically to stimulate reproduction as well as stimulate molting. So the idea is that, say, you can knock out the MIH receptor, whatever that is, we ourselves are actively engaged in trying to identify possible candidates for MIH, and we've identified actually a few G protein coupled receptors that seem to be the most likely. But the idea is, what if you knock out genetically the MIH receptor? Now we have, and the Y organ. So now the Y organ is no longer sensitive to inhibition by MIH. And so now you have an animal that is continually molting, right? So as soon as they finish one molt, they'll grow and and so basically the idea is that if you can do that, you can increase your yields from ponds, right? Because now instead of maybe having 2 crops a year, you might have 3 or 4, because the animals are growing much more rapidly. So I think that they're moving into those types of technologies that creating genetic strains that actually are inherently molting, and perhaps investing into using CRISPR-Cas9 technology to knock out the MIH receptor, for example.
Yeah.
And there you have the idea of being able to say, okay, we're just going to be knocking out the receptor, so no other physiological processes will be affected.
Yeah.
That's the idea, but usually something else comes up.
That's interesting. The last question. So I mentioned this at the beginning, climate change, and we hear in the news all the time how climate change is affecting various species of whatever, insects or things in the ocean. How are the crustacean species doing in general?
They're doing okay, mainly because they're mobile, and so they can migrate to deeper water or to maybe other latitudes. But that usually has some environmental consequences. So what's scary is, of course, the ocean has been a really great reservoir for the excess heat being produced by climate change, but the thermal capacity of the ocean is reaching its limits.
Yeah.
I mean, recently we're talking about surface temperatures of like 90 degrees. I mean, that's bathtub water, right? That's really scary. And of course, that's the surface. And if you go deeper, it's cooler. So there are refuges, as long as the animals are mobile and can still gain access to the resources they need, they'll hunker down. But that if they're going deeper offshore, for example, are they going to be accessible for fishing, right? And the changes in the Arctic are really scary, affecting cold water crustaceans like the snow crab and the red king crab. I've been collaborating with a group of fishery biologists in Norway with the red king crab, and the red king crab, or the Alaskan crab, that species was introduced by Russian Soviet scientists in the Barents Sea, so off Siberia, right? They said, Barents Sea didn't have any red king crab, and they said this is a perfect habitat for that. So let's dump some more. Let's move and dump some more red king crab into the Barents Sea and see what happens. They got established, and now the population is thriving. Right? It's now created a great fishing industry in the Arctic Ocean. But what's happened is that the population has been very successful, and the population is now expanding and has expanded over the Scandinavian peninsula, and now Norway is taking advantage of that fishery, right? And but what's happening is that the red king crab are continue to migrate down the west coast of Norway, right? And they don't really know how far it's going to go. The problem is that it's now beginning to interfere with the cod fishery off of the coast of Norway. And cod is king in Norway, right?
Yeah. They bury it in the ground, and leave it for a while, and then eat it.
Yes, lutefisk, right? Anyway, that's an interesting.
But is that because these lobsters are killing the small cod?
No, this is the bizarre thing about red king crab is that they get to be huge, right? They have the leg spans of like 6 feet, right?
Yeah,
and they because of their nice long legs, they're popular for eating, right? You can just have crab legs, right? Snow crab is also popular, but they're not as big. But so these are benthic species. They live in several into 50, 60 feet of depth on sandy bottoms, and they forage at night. They look for worms and other goodies in the substrate, and then during the day they're exposed. And so especially juveniles, because they're particularly vulnerable to predation by a variety of different ocean life like seals and even cod. And so what the juveniles do is that during the day, they all gather together and form these huge mounds that are meters high. And basically they orient their bodies to the outside. And so it's a defensive position and so they pile up and then they'll disperse at night, right? Well, during the day, that's when the cod are active, and that's when the fishermen are trawling with these big nets for cod. And unfortunately, these nets can hang up in one of these mounds, right, and tear the trawler gear to shreds. And so the cod fishermen hate the red king crab. And so the fishery biologists there were trying to figure out how do they mitigate this, and so that the two fisheries don't really conflict. And so one of the things about temperature is that these are cold water organisms, and basically how far down can they migrate and still be within its temperature tolerance, right? And so so it was a fascinating problem. So this interesting unforeseen conflict between two major important fisheries in Norway.
Yeah, interesting.
So, but red king crabs. Yeah. So in terms of we know that, but temperature is also a very important factor in molting, because these are cold-blooded organisms, and so their metabolism is related to their environmental temperature, right? So we know that temperature actually can stimulate molting. The higher the temperature, higher metabolic rates, the faster they molt, right? But at some point, temperature reaches a level that the animals can't do that as well as maintain basic body organismal functions. So that's when they reach this tipping point, and it actually inhibits molting, because now they're trying to conserve energy to maintain their organ systems, and if it's under prolonged conditions, they can die. There's some, obviously acclimation. We know that crabs and other species can acclimate to current conditions, but at some point the temperatures are going to be so high that there's no way an organism can actually acclimate and adapt to those new thermal regimes. So the bottom line is, right now it seems to be okay, but as we start seeing these consistently high levels of ocean temperatures for prolonged periods, that's going to have serious environmental effects on the oceans. Just as they are having now in terrestrial environments, these serious heat waves, right, that everyone's experiencing in the northern hemisphere. So, but tourism, is again a very important component. We find that there's adjustments as the animals are adapted to particular temperatures that actually act to modulate the metabolic rates. So there's some adjustments actually as temperatures increase. There's actually a down regulation of mTOR, because of course the higher metabolic rate is now is higher at higher temperatures. So it's kind of tweaking the thermal environment and metabolism so that proper balance and allocation of resources are made at varying temperatures, at least within their physiological range.
All right, Don. Well, I'm glad I contacted you, and it's good to see you after 40 years or whatever, about 40 years. And enjoy the last 2 years with your students, and then enjoy your retirement.
Well, thank you very much. I've really been enjoyed talking to you, and it's just molting is really an amazing process, and I didn't think it would keep me engaged for my entire research career, but it has, and been able to explore different aspects of it. So it's kind of fun.
No, it was important. Obviously, PhD work, it was an important part of my career development, and I started out like I did a master's on studying adrenal steroidogenesis with cultured neurons. Then I went to a neuroendocrine system, and then I went to mainly just studying the brain and mammals. But
Yeah,
yeah,
yeah. Well, and Gene Spaziani, your mentor, your PhD mentor, was a great guy.
yeah.
I got to know him in several NSF grant review panels. Just a marvelous person. And
Yeah.
he must have been a great mentor for you.
Oh, he was, and he was kind of like you said, the old school. He gave me some initial ideas, but kind of let me run with what I wanted. And you must know Doug Watson too, then. No?
Oh yeah,
Yeah.
sure.
Yeah.
Yep. Yeah.
And he's still working on some of these systems, mainly in insects now, I think. Yeah.
Yeah. Yeah. No, no, it's cool to see people who've worked with Gene and what they've done in their careers as well.
Yeah, Doug and I were there at the same time. He was doing a postdoc and I was doing my PhD. Okay, Don. Thanks again. I appreciate it.
You're welcome.